Hello, it's Callum Robertson here again. So this session, I'm going to cover the normal appearances, histological appearances of skin. So I've got our section from the previous video of skin. It's not an entirely normal section because on this side of it we've got an abnormality. This is a basal cell carcinoma. We're going to ignore this for the purposes of this video and concentrate on this side of the slide, which by and large is at least relatively normal. And we've got enough here, enough normal here that we've got some things to talk about. So that's the main thing. So skin, as we know, it's got a few functions. Its main function is that of a barrier. So a barrier to provide mechanical protection to the body, but also a barrier to keep out pathogens. It's also got a sensation function, so it's got the ability to sense pain, temperature, vibration, touch. It's also got a thermoregulatory function, so that is it can provide warming effects through the hair and also the subcutaneous fat, and also cooling effects through dilatation of blood capillaries through sweating. And finally, as a metabolic effect through fat storage and also vitamin D synthesis. So the overall structure of the skin is broken into three main components. That is the epidermis, dermis, and subcutis. So the epidermis is this top part here. This is the epithelial component. It's the epidermis. Underlying that, there's a dermo-epidermal junction, small, thin dermo-epidermal junction. Underlying that, there is the dermis. So the dermis is everything from the epidermis all the way down until you come to fat. And as soon as you get to fat, you're in the subcutis, or hypodermis, as it's sometimes called. And that's everything from up here. Obviously, we've got a bit of an undulating lower dermal border here. But everything below that is fat. And here we've got skeletal muscle. So once you get to skeletal muscle or fascia or something that's no longer fat, you're no longer in skin. Essentially, you're on the deep tissue below the skin. So let's talk about these in order. So let's start with the epidermis. Now, the epidermis, skin can look a wee bit different histologically depending on where you are in the body. Acral skin, for example, like the skin on the soles of the feet or the hands, can be a lot thicker. It has to deal with a lot more mechanical pressure, and consequently, it's adapted to be a bit thicker. You can also get thin skin, which you can get particularly, for example, in the abdomen. Doesn't have to deal with a lot of pressure. It's a lot thinner. And you can get, and this skin is probably from the face. Not exactly sure where it came from. It's probably from the face. Here the skin's sort of somewhere in between. So if we look at the epidermis, first of all, we'll focus on this area here because I think it probably illustrates the layers of the epidermis most clearly. So we'll start at the bottom here. So we've got different layers. The name for the bottom layer is the stratum basale, and that is just the single layer right at the bottom of the epidermis, just above the junction between the dermis and the epidermis, and it's called the stratum basale, and that's where replication happens. So these are the stem cells, if you like, for for all the cells above it that make up the epidermis. So these are replicating. I haven't. It's not very common actually to see them replicate, to see an actual mitotic figure. You can sometimes see that, but these are the stem cells. So these are all keratinocytes. That's what the name what we give for the epidermal cells. And but they have a different appearance depending on their degree of maturation. So they start here. This is where the stratum basale, or the basal layer, that's where they replicate from, and they mature upwards through the different layers to the top, to the keratin, surface keratin layer here, where they're sloughed off. And this process is a continual process of self-renewal that's happening all the time. So there's underlying this stratum basale or basal layer, there's a basement membrane, and the stratum basale is attached to this basement membrane through connectors called hemidesmosomes that fix the epidermis onto the underlying tissue, and that connects them onto the basement membrane. So above the stratum basale, there is the stratum spinosum, or also known as the prickle cell layer. So that's the area from here, from just above the stratum basale, all the way up to this granular layer, which is that's the next layer up. So it's everything in between here and here, and it's called the stratum spinosum because the cells have, or a prickle cell layer, because the cells have spines or prickles, and these are the desmosomes that actually connect the cells together. These cells are all these keratinocytes are all connected to each other, and you can see really nicely here between this cell and the one adjacent to it, these two keratinocytes. And this is the nuclei of the cells, surrounded by pink cytoplasm, and they're the cell outlines here, and you can see between them these really clearly these cell prickles. So these are the desmosomes that are holding the cells tightly to each other, and that's why that gives the stratum spinosum or prickle cell layer its name. As we continue upwards, we get to this layer here, where we're starting to see loss of the nuclei, and within the cytoplasm, instead of looking like pink like we've got down here in the stratum spinosum, in this layer, the stratum granulosum, or you've got, or the granular cell layer, if you like, you've got these little, it's kind of characterized by these little keratohyalin granules. They take up quite a lot of the hematoxylin pigment, and it makes them appear purple. So that's the granular cell layer. As these mature upwards, they undergo programmed cell death or apoptosis. You get loss of the nuclei. They're kind of dying at this stage, and then they kind of merge into the final layer, which is the stratum corneum. At this point, there's no living cellular cells here anymore, or shouldn't be under normal circumstances at least. The keratohyalin is mixed with tonofilaments to produce keratin, and this keratin is kind of the tough acellular material which provides a lot of the protection that the epidermis gives to the skin, and that's this pink stuff on the top, and that gets kind of sloughed off over time, but that's okay because it's under a continual process of renewal, and that's really probably most of there is to say about the keratinocyte component of epidermis. But there's actually some other cells here which are not so easy to appreciate at first, but when you start going looking for them, you can begin to see them, and they are the melanocytes. That's another major component of skin. We really care about melanocytes in pathology because they are the source of one of the main skin malignancies, which is melanoma. But under normal circumstances, melanocytes are visible, and if we look closely, we can pick them out. So what we're looking for here is a round cell along the basal cell and the stratum basale, but a round cell with clear cytoplasm, and I think we've got one here. So that's slightly different from the other basal cells. This is maybe a basal cell here. It's a little nucleus, but it's eosinophilic cytoplasm. And that's probably 3 keratinocytes in a row. But then if we follow it around, this looks slightly different. Round nucleus, small nucleus, surrounded by clear cytoplasm. And it may strike you as odd that it's clear because melanocytes, as we know, produce melanin, which gives the skin its pigmentation, or is responsible for giving you a suntan if you live in a sunnier part of the country or the world than I do. But the reason for this is clear is actually the melanocytes don't they have little tendrils that feed the melanin to the adjacent keratinocytes. So actually the melanin resides in their adjacent cells. So if you can, I don't know if you can really appreciate this in this blurry video, but it's quite subtle, but there's kind of brown pigment here, here, around here, and that's normal. That's normal to see, because the melanin is providing protection against UV light, natural protection, and it's normal to see a little bit of melanin here along the basal layer, but usually not within the melanocytes themselves, usually in the adjacent keratinocytes. So if we follow this round, we can maybe see some more melanocytes. And there's another one, brown nucleus, clear cytoplasm. Keep following it round. Maybe we can see some more. We're kind of going down a hair follicle here, so we wouldn't expect to see melanocyte. Here we are. Here's another melanocyte, round nucleus and clear cytoplasm round about it. So that's there's another really good example. You know, that's definitely not a keratinocyte. That's definitely a melanocyte. There we are. So that's that's melanocytes. There are other cells in here. So there are Langerhans cells, which are histiocytes, or part of the monocyte macrophage antigen-presenting system cells. This might be one. They're further up usually. They're interspersed with the keratinocytes, and they also have clear cytoplasm. Sometimes they have a wee bit of a crinkly nucleus, clear cytoplasm. So this might be one, for example, and it doesn't have I can't see desmosomes necessarily around this cell, so it could be one, but you can't really identify them very accurately on H&E. And the other main cell type that you've got you can see Merkel cells. I don't know what they look like. They're indistinguishable on H&E, to be honest. I think you need immuno to really pick them out, but they are there, and they're I think they're help in providing touch sensation. We don't really come up. We don't really need to identify them in normal circumstances. They can be responsible for tumors in rare circumstances, but in general, we kind of largely ignore them. So underneath the epidermis is the dermis. So that's the bit from the bottom of the epidermis all the way down to until the subcutis begins. And the dermis is made of two main parts. There's the papillary dermis and the reticular dermis. I'll see if I can find a spot. So the majority of this is reticular dermis. Papillary dermis just is where the dermal papillae, that's these little tongues of dermis that project up, that interlock with the downward projections of the epidermis, which are the rete ridges of the epidermis. So yes, this most superficial part of the dermis is the papillary dermis, so-called because of these papillae. And it contains the same kind of stuff as the reticular dermis. There's lots of collagen and elastin and blood vessels and so on. The kind of nature of the collagen and elastin fibers tends to be a bit finer in the papillary dermis, whereas these big blocky collagen fibers you tend to just see in the reticular dermis for the most part. The other way you can tell apart is there's a superficial vascular plexus. So here's a blood vessel running along. That's probably running along the vascular plexus, which separates the papillary dermis above it with the reticular dermis below it. But sometimes it's not quite so obvious to tell exactly where the junction is. And yeah, below that is a reticular dermis. I don't think there's a great deal to say about that. I would point out that this is not entirely normal, actually, this skin, because in here you've got this. See these kind of aggregates of this pale pink stuff. This is actually elastin, and it's not usual to see these big aggregates of elastin like that. You might see a few elastin fibers under normal circumstances, but this is too much, and this is actually sun damage. So this is probably from someone's face, and it's just maybe you know, they've just had a bit of sun damage. That maybe, which is normal to see on the face. You know, once you get to a certain age, we'll all have a degree of sun damage, and it makes a kind of sense as well when you think that this person has developed a basal cell carcinoma, which is heavily associated with sun exposure. So it kind of all ties together that they would have a bit of sun damage within their dermis too. Okay, so we've gone through the epidermis, the dermis. The subcutis is really just made of subcutaneous fat. So this is made of adipocytes. There's really very little to say about adipocytes. Adipocytes are quite big. They're balloon-like. They've got nuclei over to one edge, and the cytoplasm is completely cleared because we use alcohol as part of the processing of these, and alcohol dissolves out the fat, leaving these big balloon-like spaces. And these are adipocytes. These are normal adipocytes. You can see a slight variation in size and shape in adipocytes, and that's okay. This is just normal fat. It's very quite unexciting, to be honest. However, there's some other things here that we haven't talked about yet. So the first I'll talk about is this stuff. So this is a pilosebaceous unit made of hair follicle here and sebaceous glands on either side of it. And one last thing to make up the pilosebaceous unit is the rector pili muscle, which is here, a bit of smooth muscle, which is responsible for changing the hair follicle from the kind of horizontal position to the vertical position in response to, which happens as part of the thermoregulatory system of the skin. So the first part of this that we'll talk about is the hair follicle. Now, we've not really, unfortunately, got a great section here to show you exactly the histology of the hair follicle, and I'm by no means an expert on hair follicle histology, but I'll just give it a quick outline. So at the bottom of this is where the hair follicle, the actual hair shaft, which is this bit in the middle, is produced. Sometimes you can see a little indentation at the bottom here if you've got a better section, and that's the dermal papilla, which is which you might see, which is quite vascular and provides a blood supply for the follicle here. And then sometimes at the bottom you can see it nicely that the bulb dilates a bit at the bottom here. Unfortunately, we've not got a good section of that. But the hair follicle itself has got an outer root sheath, which is this kind of modified epidermis with clear cytoplasm. That's how you identify the outer root sheath. An inner root sheath, which has got these kind of pink granules, quite eosinophilic granules. And then within that, in the center, there is the hair shaft itself, which if you could see a good section, you would see it coming up. Often it's made of keratin. It's often quite, you can see it has this kind of brown look. It can look quite refractile. And that would, if we had a good section, you would see that coming up here and then emerging from the surface. And that's pretty much all I have to say about the hair follicle. Associated with that are these structures. So these are completely different. These are part of the pilosebaceous unit. These are the sebaceous glands. These are associated with the hair follicle and open about a third of the way up the follicle or a third of the way down the follicle. And they secrete their secretion, their sebum, into the hair follicle, and then it emerges out onto the surface skin here. So these are the cells of the sebaceous glands, and they are known as sebocytes. You can see the sebocytes, you can see the nuclei, and they're surrounded by this kind of bubbly cytoplasm. These have an oily-based secretion, which is fat-based, and it's dissolved out, again, a bit like the adipocytes, it's dissolved out during processing, leaving these clear spaces. So it's lipid within these vacuoles that would be within these vacuoles if it hadn't been dissolved out. And these are an example of holocrine secretion. So holocrine just means that the cells themselves produce this secretion within their cytoplasm. Then they mature up a bit like in the skin, in the epidermis, they mature up, they die off under programmed cell death, apoptosis. You can almost see this in action here because you've got viable cells with viable nuclei down here maturing up, and then here you kind of lost the nuclei, and that's as these cells are dying off, and then as they die, they rupture, and the cells release their lipid, their secretions up here into the hair shaft, and you can see the hair follicle. I'm sorry, lumen, and then you can see how that secretion would continue up here onto the skin surface. So you can kind of see that in action there. It's quite nice. So that's an example of holocrine secretion where the cells die off to release their secretory product. So that's probably all I'll say about sebaceous glands. The other main gland type we've got here are the sweat glands. And there's an example of one, and there's an example of another one. These are not associated with the pilosebaceous unit. These are completely distinct from the pilosebaceous unit. And these are usually found, as in this case, within a kind of superficial subcutis. And they look like a lot of little glands, but actually they're all connected in this one big coil, because remember, this is a two-dimensional section through a three-dimensional structure. So this would come out, this tube would come out towards us and curl back around and then go back in here and then out here and in here and so on and so forth. So it's a coiled structure, and these are eccrine sweat glands, as opposed to apocrine sweat glands. So I know this because these are apart from their appearance. It's a good chance if you see sweat glands, they are eccrine, because apocrine sweat glands only occur in special settings, but eccrine sweat glands are pretty much throughout almost the whole body, skin, and almost all sites of the body. These produce a watery kind of serous secretion, and it's made of these are made of secretory tubules, and also, or some of the tubules are secretory, some of them are just for transport, and I don't think it's very easy to tell which of the tubules is secretory, and which of them are ductal structures that will, the ducts will lead to the surface. But the secretory ones are supposed to just have one cell layer. So I suppose that maybe has one cell layer there, and it has a surrounding myoepithelial layer. Now, myoepithelium, it just is epithelium that has the ability, it's called myo because it has the ability to constrict a bit like a muscle cell, and it can squeeze. The point of that is to squeeze the secretory product through the lumen towards the ducts. So they may be one cell thick, so that's maybe the secretory component. And here, this ductal component is supposed to have two cells. It's supposed to be bilayered. So maybe that's got one layer and then another layer beyond it. And the way I can usually tell the ductal cells is because they've got this more intense pink luminal aspect. So this is the ducts. So it's producing sweat in this coiled glands, and then go up through the duct all the way up here, up to the surface, and it will open up through an opening in the epidermal surface up here. So that's eccrine sweat glands, and they are eccrine as opposed to, they're not holocrine like the sebaceous glands. These don't die off, these cells. These just secrete the secretions into the lumen. Apocrine sweat glands would look maybe broadly similar to this. They would be a bit more dilated, and they have little blebs that bleb off, little almost like teardrop-shaped little pink secretions that bleb off into the lumen. They look a bit different, but unfortunately, I don't have a good example of that. Maybe you can look at a book to see them. So that's pretty much everything. We covered a lot of the structures here in my previous video anyway, and that's pretty much all. That's pretty much all there is to say about skin. There's one last thing I would say, that sometimes we talk about Clark levels in the skin, so I'll just quickly go over them. By and large, Clark levels are in relation to the to melanoma, and what they denote is the level of the skin that the melanoma has invaded to, i.e. the deeper the worse it is, and so Clark is given, the Clark levels are 5
1 to 5, depending on how deep through the skin it's gone. And occasionally people will use Clark levels as a descriptor for other tumors other than melanoma. So I'm just going to go through them quickly now. So Clark level 1 is within the epidermis, I'm sorry. Clark level 2 is within the papillary dermis. Clark level 3 is the papillary reticular dermal junction. Clark level 4 is the reticular dermis, and Clark level 5 is in the subcutis. So obviously 1 to 5, 5 being the worst, 1 being the best in terms of the least, the least invasion that there has been. But not really relevant for normal skin, but it is something that you might see, so it's useful to have an awareness of that. So that's it. That's a quick run through of histology of the skin, with hopefully a bit of a practical focus, and hopefully that gives you some revision of the normal structures within the skin. I'll be doing another video shortly. I think the next one I'm going to do is some GI histology. So until then, I'll speak to you soon.